Before to move on flash talks, I want to remember that poster evaluation is going on also during the cocktail time. Then I recommend to authors to be in front of their works, paying attention to the questions of the jury. Now, I said, we are starting with the flash talks. The first speaker is Kilian Abellaneda, that belongs to Brain Health and Neuromodulation Group, and he's presenting us Purpose in Life, a psychological factor promoting cognitive resilience and neuroprotection.
So, good afternoon, and thanks to the organizing committee for having me today. My name is Kilian Abellaneda Pérez, and I am a postdoctoral researcher here at the University of Barcelona. And within this session, I will present our recent data on which we aim to investigate the relationships between purpose in life, a common age-related brain change as the accumulation of white matter burden, resting state functional connectivity, and cognition in a sample of healthy middle-aged individuals. So, it is well known that increased life expectancy represents one of the biggest transformations of age structure among contemporary societies. This represents an important accomplishment, but also poses an enormous societal and health challenge, as advanced age is a principal risk factor for many highly prevalent and disabling disorders, including Alzheimer's disease. In this context, and despite intense research efforts and large investments from public and private sources, disease-modifying agents to contract age-related cognitive impairment remain largely elusive. However, it is worth noting that recently developed drugs and therapies may suppose a promising game changer on the field. Nonetheless, identifying modifiable factors that may promote brain health, reserve, and resilience alongside the lifespan is still paramount. So, in Alzheimer's disease, education and occupation characterize the classic cognitive reserve proxies, which can be potentially targeted and promoted over the life course, providing resilience to age and disease-related brain changes. Nonetheless, the importance of psychological factors is being increasingly recognized as their operating biological mechanisms are elucidated. Within this context, purpose in life, included in the RIF prevalent model of psychological well-being, is one of the most promising psychological constructs. Purpose in life refers to the sense that life has meaning and direction, and that one's goals and potential are being achieved or are attainable. Purpose in life relates to better cognitive and affective status, and it may delay cognitive decline and the onset of cognitive impairment, thus potentially having a role as a reserve mechanism against AD-related pathological changes. However, and despite evidence in this line is increasingly compelling, the neurobiological mechanisms underlying beneficial effects of having a purposeful life are not currently well understood. Hence, in the present study, our main aim was to delineate the functional brain mechanisms that could underlie the expected protective effects of purpose in life in the face of age-related brain burden. So, regarding the methods, data was obtained from 624 middle-aged adults from the Barcelona Brain Health Initiative. This is an ongoing longitudinal cohort study investigating the determinants of brain and mental health in middle-aged individuals. We later conducted analysis focusing on the extreme groups, as previously known in the field, the quintile 1, named lower purpose in life group, and the quintile 5, named higher purpose in life group. These two groups shaped our sample of interest conformed by 246 individuals. Purpose in life data was analyzed in terms of cognition, episodic memory, working memory, and executive function composites, white matter lesions as a highly prevalent structural brain change in maturity, and resting state fMRI, with a measure of system segregation. This metric was chosen because it condenses in a single estimate the functional connectivity of within and between brain network nodes. So, let's go to the results we obtained. First, we observed that age, neuropsychological status, and white matter lesions did not differ between higher purpose and lower purpose groups. However, we observed a significant group interaction in the association between executive functions and white matter lesions. More specifically, greater white matter lesions were negatively associated with executive functions only in the group with lower purpose. When considering resting state fMRI data, we observed that subjects in the higher purpose group showed lower system segregation on the dorsal default mode network, the dorsal DMN, than lower purpose subjects, which indicates lesser segregation of this network from other brain circuits. Precisely, higher purpose individuals had greater internetwork connectivity between dorsal DMN nodes and the rest of the brain on the functional couplings depicted on the left figure. It is worth noting that out of the total connections that emerged in this analysis, the frontal cortex, the hippocampal formation, and the mid-cingulate region entailed the more relevant nodes. Conversely, subjects in the lower purpose group exhibited more connectivity than higher purpose individuals across DMN-like regions, particularly those involving connections between the dorsal DMN and the ventral DMN, as can be seen in the right figure. Of note, no differences were detected on the whole DMN between network connectivity with other brain circuits, neither in the connectivity within DMN nodes. Hence, our system segregation results were likely driven by a specific dorsal DMN nodes with enhanced internetwork connectivity with other brain regions. Finally, we investigated whether resting state fMRI might differentially subtend cognitive function for a given brain burden estimation as a function of purpose in life group. Considering the previously identified functional couplings, we detected two functional connections positively associated with executive performance on the higher purpose group. Both connections involve the mid-cingulate region of the dorsal DMN and two other cognitive brain systems, the posterior salience and the precuneus network. No associations with cognition were observed in the lower purpose group. So, as a general conclusion, we can state that present data expand previous findings on advanced age and pathological aging as Alzheimer's disease, revealing that having a strong sense of purpose may confer cognitive resilience already in middle age. Further, it was also observed that individuals in the higher purpose group had greater internetwork connectivity between specific dorsal DMN nodes, which correlated with cognitive performance. This may represent a potentially neuroprotection mechanism related to greater purpose in life. So, thank you, and here's my email address where you can reach me out for any comments, suggestions, and further discussion about the presented data. Thank you very much.
Thank you, Kilian, for adjust to time very well. And now it's time for 2 minutes questions. There is some question. Yeah.
Thanks. It was a very nice talk. So my question is a little bit about the process of how you designed the study, in the sense that, to me, it's a really, really interesting, but also very specific measure. So I wonder whether it was something like, I know from the literature that purpose in life needs to be related to my other measures and to my brain measures, or whether it was more like there are plenty of candidates that could have this relationship and the purpose in life was this one that stood out.
Yes, this study actually it's part of the Barcelona Brain Health Initiative that is a longitudinal prospective cohort study, and there is a lot of potential lifestyle factors that may predict cognitive and brain health and mental health. So we have a factor that it's more related with meaning in life. There is the purpose in life, sense of coherence, engagement with life. But previous studies, particularly in Alzheimer's disease, focus on purpose in life. So we selected purpose in life because of these previous studies. But also we are interested on if this is not a relation that it's only in the pathological brain, but we can find something before. We can find something in this line before the pathological conditions. So, in a sense, the main idea is the purpose in life may confer some reserve or some cognitive resilience, so the brain is less dependent on the structure to function correctly, in a sense.
Some question? Okay, what do you? This study is a part of a big study and it's prospective also. Then you can follow on these patients to a period of years to follow the possibility of onset of dementia in which these patients that have low rate of purpose of life.
Yeah, actually, this is the main idea of the project. So start from the health and go to the potential pathology, so it have a salutogenic perspective. So yeah, this is the main idea. The founding we'll see, but the main idea is this one. Yeah.
Okay, thank you very much, Kilian. We move to Julia Solana. Julia Solana belongs to the group Mechanistic and Therapeutic Approaches in Neurodegenerative Disorders, and she presents us the talk entitled Neuron-derived extracellular vesicles modulated synaptic plasticity.
Okay, thank you. Good afternoon, everyone, and thank you to the committee for inviting me to present our last results about neuron-derived EVs and synaptic plasticity. Neurodegenerative diseases are a very heterogeneous group of diseases characterized by a progressive loss of brain function. Neurodegeneration shares common hallmarks, and among all of them, there is a dysfunction of synaptic plasticity. Neurodegeneration starts then in an alteration in the neurotransmitters that affect synapses, neurons, impairs circuits, and finally ends up affecting the whole neuronal networks. Patients suffering from these diseases show an early synaptic deficiency which highly correlate with cognitive deficits. So we have asked how could we prevent these synapse losses that appears at the early stages of the disease. To do so, we have focused our research in extracellular vesicles that are vesicles formed by a lipid membrane that have been widely described to be involved in intercellular communication. I don't know what, yeah. So extracellular vesicles, or EVs, contain proteins, RNAs, and lipids that are biologically active when they reach the recipient cell. There are different types of EVs distinguished by size, biogenesis, and cellular origin. And in the context of the brain, they ensure the physiological functions, allowing proximal and distal interactions among cells. In this project, we have focused on the study of extracellular vesicles derived from neurons, as there are some studies that point toward a regulatory function of synaptic processes. But how do neuron-derived EVs modulate synaptic plasticity? To answer these questions, we have isolated EVs derived from neurons using a protocol of sequential ultracentrifugation. We have characterized them using different techniques, and we have used them to perform treatments to other neurons for 24 hours, and we have assessed the effect of this treatment in the recipient neurons. So the first thing we saw was that our EVs fractions were enriched in many synaptic proteins, shown in green. And we went on to analyze how these EVs were internalized by neurons by labeling them in red. And we observed that EVs were internalized at the level of the soma and also in dendrites, but also even specifically in dendritic spines, suggesting that these EVs might be having a direct effect over synapses. So next thing we did was to assess which endocytic pathway was involved in this internalization. So to do so, we used two different inhibitors, chlorpromazine, which inhibits clathrin-dependent endocytosis, and dinosaur, which inhibits both clathrin and caveolin-dependent endocytosis. So as you can see here, without these inhibitors, we observed the normal uptake, but when inhibitors were used, we considerably reduced EV's uptake by neurons, suggesting that clathrin-dependent endocytosis might be a major pathway of endocyting these EVs by neurons. So the next thing we asked was whether these EVs were having this direct effect over synapses after the treatment. So we treated neurons with the EVs, and we assessed the spine density. And as you can see, after the treatment, neurons display a higher spine density, and moreover, they had more spines that were active as a readout of clusters PSD95 and big LUT. So the next thing we asked was whether this effect was only localized to spines or was also visible in the whole neurons. To do so, we took the total neuronal lysates treated with EVs and we performed a Western blot analysis to assess the levels of PSD95 and bGlut. As you can see, PSD95 levels were also increased when we were looking at the total neuronal lysates, and we see tendencies in the case of bGlut1, suggesting that although the effect is somehow visible in the whole neurons, it may be more localized to synapses. So the next thing we asked was which signaling pathway was involved in the synaptic effect of the EVs. And to do so, we focus our attention in the mTOR pathway, as it's a pathway that has been widely considered to be involved in spinogenesis and synaptic processes. And specifically, we focused on a substrate that is the S6 ribosomal protein, which is a component of the 40S ribosomal subunit and has been involved in protein translation. So this is also considered to be a crucial regulator of protein synthesis locally in the synapses. So what we saw was that after treatment, neurons had more activation of this protein, suggesting that the synaptic effect might be mediated by the local protein synthesis derived from activation of the S6 protein. So next thing we wanted to assess was whether EVs could have also an effect over neuronal networks. To do so, we measure calcium entrance in our cultures in collaboration with Mercè Massana and Jordi Soriano. So we got the raster plots in both conditions, and summing up vertically these raster plots, we obtain the coactivation sites. So first of all, we assess individual activity, and we, as you can see here, we got no differences in the number of spikes and neither in the interspike intervals. So we went on to assess collective activity, in which we didn't see differences either in the activity of the network or in the interburst interval. These results indicate that neuron-derived EVs might be mediating structural changes in synapses, providing them with all the machinery necessary to be active in response to an input, but do not change spontaneous activity of these neurons. So finally, we wanted to assess whether these EVs could have a trophic effect over an altered neuronal culture. So to do so, we use a model that is called a nutrient deprivation model, in which you take out from the medium of neurons a nutrient that is the B27, and we use this model to treat with EVs and see what happened. We use two different concentrations of EVs, and we assess neuronal morphology after the treatment. So we didn't see an affectation of this nutrient deprivation in the case of primary dendrites, nor in the case of endpoints, but we did see that this nutrient deprivation affected intermediate branches, and we totally recovered the normal arborization after EVs treatment at the highest dose. So with all of these results, we can conclude that neuron-derived EVs modulate synaptic plasticity by consolidating excitatory synapses by S6 ribosomal protein activation, and that these EVs could act as neuromodulators to palliate both synaptic and morphological neuronal impairments in neurodegeneration. So I want to thank my group and my collaborators, and to all of you for listening.
Thanks very much, Julia. We have time for one or two short questions because we are late. Silvia, here.
Thanks, Julia, for this nice talk. So my question is regarding the content of these EVs in different types of neurons. I mean, because you are just making a general conclusion saying that can be affecting, but did you look for different contents of EVs from different cell types and you try to mix different cultures or something like that?
No, all my results are done with cortical neurons only because we wanted to see this modulation of excitatory synapses. So it's why we decided to use these neurons. But I think that is very interesting to compare the effects of, I don't know, maybe hippocampal neurons, striatal neurons. Yeah, but we haven't done that.
And okay, thanks. And it's known if glia cells also release these EVs?
Yeah, EVs are released by almost all type of cells, so yeah, they have a lot of effects as well.
So
yeah.
I don't know if you will keep time, but try to just make co-cultures of astrocytes or microglia or whatever with the neurons and try to see if there's any relationship with these EVs?
Yeah, it actually is very interesting because it's seen that glial EVs in neurodegeneration promote this inflammatory context. So I think that is very interesting to look the interaction of glia and neurons, communication between EVs. Yeah.
Okay, thank you very much, Julia. We moved on to the other. Álvaro? We move to Álvaro González, that belongs to the Addiction and Dual Disorders group, and he will talk us about cognitive performance in patients with dual disorders, the influence of the type of severe mental illness.
Perdón. Well, hello, everybody. My name is Álvaro González. I'm a psychologist. I work in the psychobiology department of University of Barcelona, and I'm going to talk about dual disorders. These dual disorders are when people that has a severe mental illness, that can be schizophrenia, bipolar disease, or depression, and are also consuming drugs. This long title has the information about this. So, a little introduction. This is SUD is substance use disorder, and MI is severe mental illness. So we know that at least 50% of the people that are diagnosed that has this diagnosis share the diagnosis with the other group. And also we know that cognitive functions works as an endophenotype of these diseases. So for example, in schizophrenia, we know that they have shorter speed, they are faster. In the case of bipolar, we know that they score lower in executive functions, and in case of depression, we know that they do lower things. Also, in previous study, most of the studies compare dual disorders with severe mental illness and not differentiating because of the severe mental illness. So what we did? We took a sample of 150 males, 50 males for each group. They were all between 18 and 55 years old, within 3 to 12 months abstinence, and they were all stabilized and under treatment. We do a first collection of data and psychotic symptoms with the STID, the DAST, the drug abuse screen test for seeing the severity of the addiction, and in case of the psychotic symptoms, we use the PANS in schizophrenia to evaluate the positive and negative symptoms. In the case of bipolar, we use the Young Mania rating scale, and in case of the depression, we use the Hamilton, which is a well-known scale. For the neuropsychology part, we do assessment battery with 2 subtests of the WISE, vocabulary to establish a premorbid IQ, digits to see the attentional spans. In the case of the memory, we use the RALP. I know that Valenzuela already told us about this test. It's a list of words that is being repeated for 5 times, and you can see the learning curve and memory process, such as recall, recognition, and codification of the information. To see the problem solving and cognitive flexibility, we use the Wisconsin card sorting test that, as you can see here, is the image at the top right. The subject has to choose in which pile of cards has to go a card, and this criteria changes in the test. So you can see how the subject adapts to this new criteria. To study planning or planification and problem solving, we use the Tower of Hanoi. Then with this image, I think you can understand very well how it works. Also to see the speed and cognitive flexibility, we use TMT-A and TMT-B, which is an easy test to just match numbers or match numbers and letters. All of these tests, excluding the Tower of Hanoi, had scales. This means that we can compare the results of each test with a normal population, all in set scores. That made us able to get a global setter score about how they perform in all of these tests. So results. I'm going to start talking about the normative results that we found, and also with no difference among groups. This was the case of vocabulary. We didn't find any difference in the premorbid IQ. We also didn't find any difference in digits and TMT-A and Tower of Hanoi. I have to say that in Tower of Hanoi, we cannot say if they are normative data because we couldn't find any scales on Spanish people. In the case of TMT-B, we found normative data in all of 3 groups, but we found that bipolar group did the worst. Being schizophrenia in the middle, but really worse compared to depression. In the case of Wisconsin, we had some strange results. We got normative results and no difference among groups, but we had some really high scores. I want to say that a 0 set score would be the normal score, like average people. From above 1 point is one typical deviation from the average people. So 1.5 is really high. We found that these subjects had a performance much better than the normal people. I will talk about later this. So in the case of the memory test, the RALPH, we can see clearly in this image how the schizophrenia group, is the yellow one, goes worse as the test goes. We can see that all of them score lower than average, but only we can say that it's affected if it is under 1.5 point the scale. So you can see here how the schizophrenia group goes like being tired of the try or the test. In the case of the cognitive domains that we have studied with taking into account all of the tests that we have passed, you can see here also very clearly how the schizophrenia group had a worse performance. I have to say also again that all of these tests were under the average. The results were under the average people. You can see here that in the case of attention, learning, short-term memory, recognition, and in global performance, the schizophrenia group did much worse than the other 2 groups. And all of 3 had lower results than average people, but we cannot say that this is affected unless in the schizophrenia group. So to end up and sum up things, we can say that the schizophrenia group did a worse performance in all of the tests. We can say that they had affected attention, memory, and learning. We don't know if this could be explained by the cognitive fatigue. They get tired, as you could see later before, or it could be something related with the pharmacology of this group. We know because of the existing literature that this group takes more drugs, more medical drugs than the other groups, and this could be affecting the results. In the case of bipolar, we have seen worse results in TMT-B, making it that suggesting the possibility that they had lower cognitive flexibility. This also was a good difference between the depression group. The depression group is most of the time mixed with this group in the results. So this could be a useful test to differentiate these 2 groups. In this case of Wisconsin, as I have said before, we haven't found any difference. So we don't know if this is a good test to discriminate in this kind of people. So we should be taking into account this. For ending up this flash talk, I want to say that this is the first approximation of the cognitive performance, trying to establish a profile in patients with dual disorders according to the diagnosis of the SMI, not of the drug. This also works as a weak point for us because we didn't separate the consumers because of the drug, because they were taking many drugs. And we also have to say that our strong point was to have all the tests, or almost all the tests, in Z-score, so these results can be compared with other investigations going on. Thanks for this, and that's it.
Thanks very much, Álvaro. Some questions or commentaries about the presentation?
So it's just a curiosity that you really point in the last slide. So why only males in your study?
Sorry, I was short on time and I didn't want to, okay, but I will tell. Well, there's one main reason that most of the people that are on the centers that we go to take the data are males, but this is a thing that we are changing now. Currently, we're doing some new investigations trying to take into account more of the female perspective, but this also works. We want to get into the women because there's a lack of studies about this, but it also gives strength to the data because we can say that there is sex or is not explaining this difference that we found.
And the second question, when you are saying polydrug consumers, you are meaning medical drugs?
No, I was saying illegal drugs, but we cannot separate if they are only drinking alcohol or they are doing just THC. Most of them are just mixed.
You don't separate, I mean, you stratify the patients in terms of the drug that they consume or not?
It's hard to separate because they go to one, then go back, then to another one, so, but we'll try to figure this.
Okay. It will be important the point out of Silvia, because the stratification by drugs that affect to the behavior, to the learning, and the alcoholism, etc. I think it's very important to extract the work. Okay, somebody, yeah, no. There are not questions in the chat. Then, with this talk, we finish the scientific part of the Congress. I and the organizing committee and I want thanks to the speakers for their brilliant and inspiring talk. And now we start the social and networking part of the Congress with the cocktail. And I remember again that the poster evaluation is ongoing now. Okay, thank you very much, and we see with a cup in them.